Um, Hi, my name is Suhas. I'm with the Center for Genome Architecture at Baylor College of Medicine, Rice University. And I'll be telling you a little bit today about how we're using Power8 at Rice University to accelerate our workflows and learn kind of really basic biology about how the genome is organized. So the basic problem we're interested in is the problem that if you take out the DNA out of a cell and stretch it, it measures roughly 2 meters long, but it fits inside the nucleus of a cell that's only microns wide. So this is kind of many orders of 6 orders of magnitude of packing. But yet the cell manages to do this without tangling the genome and access precise pieces of it at any given time. So how this problem is solved in vivo is of utmost importance to understanding how the body functions. But yet, despite this, the importance of this problem, while we know a lot about how whole chromosomes fold and the kind of double helix structure of DNA, how this is packed at the middle scales still remains mysterious. So how do we go about approaching such a problem? So one approach is to use a technology called contact mapping. And it's useful to use a Facebook analogy for this. The clickers. OK, there we go. So let's say I was on Facebook, and because I'm still waiting to get access to my new Power8 cluster, I have some time to kill, and so I decided to look through Homer's Facebook photos. And so maybe I see some of Homer alone, maybe some with Homer and Marge, Homer and the kids, maybe some other people make an appearance. And so now I've wasted a ton of time, so I need to justify doing this. So I decided I'm going to do some simple analysis. And so if I count the number of times any pair of people appear in the same photo, I can put that into a matrix, and immediately you can see some structure appear. So if you look, you can see that there's a square in the middle that's enriched over the rest of the map that kind of tells you the structure of Homer's family. And then Homer and Marge appear together in more photos than the Homer with the kids, so you can see that enrichment as well. So basically, if I had a Facebook for genomes, maybe I could pull some structure out of that. And thankfully, such a technology exists. It's called Hi-C. And the basic idea is we take the genome, we freeze it, we basically glue it together, we cut it into millions of pieces, and we mark the ends, the free ends of the fragments so that we can pull down, and then we connect them so that we can pull down these chimeric molecules that represent 2 pieces of DNA that were close together in space. And so if you just look at one of these snapshots, it doesn't really tell you very much. But if you look at millions or billions of these, you can start to put together a map of the 3D genome. And so when we do this, now instead of each pair being a pair of people in the photos, it's a pair of positions in the genome. And so you can fill a matrix, just like we did with Homer's photos. If I zoom in on a particular chromosome, now, anytime I see something that's more red, that indicates that those 2 pieces of the DNA were closer together in space than other pieces of DNA. So if I look at the interactions of, say, position A with itself, you'll see that it's very red, which makes sense because things that are close together in 1D are close together in 3D. If you look at something a little bit farther away, A and B, it's less red. A and C, even less red. But that takes us to the first kind of computational challenge, which is that I'm taking these chimeric molecules of DNA, but that doesn't actually, when I read off these molecules, I don't actually get the position of the genome. I get just, when I put it on a sequencer, I just get text that I then have to align back to the genome and figure out their positions. And this is not a trivial computational problem. If you were doing it just once, it might be fine, but we have to do it millions or billions of times. So to kind of set the stage a little bit, I'll tell you a little bit about our hardware setup at Rice. And so we have 8 Power8 systems for a total of roughly 1,500 virtual cores. 2 of our nodes are high memory nodes with a terabyte of RAM for high memory applications, and then 2 of them have K40 Teslas. Actually, this is a little bit out of date. They just got upgraded to K80 Teslas. And so I'll tell you a little bit later about some of the applications that we're using GPUs for to do some analysis on very basic biology. We also have 4 JBOD disks for a total of 660 terabytes of disk space, scalable to 1.4 petabytes, as well as a 20 terabyte flash array for I/O intensive applications. And we have a 56 gig InfiniBand connection between the disks and the Power8 systems. We're running Slurm over Ubuntu and Red Hat Enterprise Linux. All right, so we have this awesome Power8 system. And just to give you a scale of this problem, we've generated our highest resolution maps of the 3D genome have 13 billion reads, and we're generating billions of reads every month. So to give a sense of scale, that's nearly 200 times as much data as the entire Human Genome Project generated, and we generated in half the time, and we're continually generating it every month. And so Power8 has enabled us to do roughly 700 alignments per second per core over the 192 cores of the Power8 clusters, is 50% faster than our previous clusters. So now we can do these kind of alignments in hours, where before it would take us much longer. So using that, we can actually build maps of the 3D genome. And so our most recent, highlighted in yellow, we can basically look at how the genome interacts with itself on kilobase resolution, which is basically 1,000 base pairs of DNA. So imagine cutting the genome into 3 million pieces and figuring out how each of those pieces interact with each other. Great, so we've built this map. What does it actually tell us? So one of the fundamental problems in biology is we have our own spooky action at a distance problem, which is that there are switches, these regulatory elements, enhancers, that control genes, that turn on genes and kind of control the functional state of the cell, but they're separated by a distance. And actually, the physical proximity leads to the mechanistic action. So mapping these loops is pretty fundamental to understanding how the genome works as a mechanism, but we don't actually have that. We haven't actually done that. So with our new maps, we can actually translate this problem of looking for chromatin loops into looking for enrichments in a contact map. So now if you take a loop position and then move in any distance in either way, you'll kind of decrease the enrichment. So we're looking for these focal peaks. We run into another computational problem now, which is that the map we're looking at is a 20 gigapixel image. So this is like trying to find features that are the size of a city block on the entire Google Earth image. But we have the advantage that it's embarrassingly parallel. So we can implement this on GPUs and speed up this entire problem of looking at every pixel and comparing it to its local background to find enrichments. And so when we put it on GPUs, we get a 200-fold speed up, and we can take this problem that would have taken us weeks and make it finish it in hours. So we map these loops across the entire genome. They're reproducible and across different high-C experiments. We can look at them under a microscope and see them as well. The loops actually pinch off parts of the genome and set up the intervals of DNA interact with each other more often than you would expect by chance. So they're actually setting up these regulatory neighborhoods in the genome, just like the one big happy family. And like I said in the beginning, our main goal here was to figure out this regulatory network inside the cell. So if we compare these maps across different cells, these annotations of loops, we can actually see when loops appear in different cell types, they turn gene expression on. So using Power8 and using that to map the loops, we can actually start to decipher the gene regulatory network of the cell. Clickers. There we go. So what's actually forming these loops? So when you look at the anchors of these loops, you see that there's this particular protein called CTCF, and it recognizes a particular sequence of DNA that has an orientation. And so we see that actually at either end of the loop that the orientations are flipped so that they point towards each other. Kind of like this. So we have loops, and then at the base of the loops you have these CTCF sites that point towards the inside of the loop. So this kind of hints at the mechanism by which these loops form. So you might imagine that 2 pieces of DNA were just kind of wiggling around in space until they come into contact. But this model actually has a lot of problems. So one, it doesn't explain why you see this kind of orientation rule, because if they were convergent or divergently oriented, you wouldn't, the loop would have no way of knowing that. It doesn't explain why loops tend not to overlap in our annotation of loops. And it doesn't explain why we only see loops between the same molecule of DNA. We don't see loops between different chromosomes. So we actually came up with this other model of DNA where basically a complex binds to two very nearby pieces of DNA and starts to slide and extrude a loop, which satisfies all the properties. And so now we can actually model this. So that brings me to the next kind of how Power8 is actually helping us advance the science here. So we can use molecular dynamic simulations. And if you could actually play the movie here. We can use molecular dynamic simulations to actually model this process of extrusion. But these simulations are extremely computationally intensive, and each kind of simulated contact map requires hundreds of simulations. So when we put this on GPUs, we can get a tenfold speed up and model different regions of the genome much more quickly. And here you can see that this complex, as it slides along the DNA, actually sets up the red and blue segments of DNA where they were tangled in the beginning after this process of extrusion end up kind of being segregated into insulated domains. And so this actually enables us to do this actually enables us to kind of skip the experiments altogether. And so rather than having to use this expensive DNA sequencing to figure out what the 3D structure of the genome looks like, we can just use now molecular dynamic simulations and recreate these maps without having to even sequence any DNA. So the top is in silico simulation, and the bottom is what you would get if you did this whole DNA sequencing experiment to figure out what the structure looked like. And you can see they look almost identical. And so this allows us to now even do fancier things, which is we can predict the changes. So there are a lot of diseases in which you might expect the genome to be misfolded. And so one goal might be to kind of correct the misfolding and to go in a targeted way and change the folding of the genome. And so now we have this tool to actually go in and see, by using simulations on the Power8 cluster, we can actually see what those changes are going to result in before we actually do it. And so now here, this is just before we make any changes. The left is a simulation, and the right is the result of the experiment. If we delete one of the anchors of the loop, you'll see that our simulation predicts that loop and that domain will disappear, and that's the same thing we see with the experiment. We can do this with other kind of deletions of loop anchors, with a whole number of them. And every time, exactly what's predicted by our simulations ends up what happens when you actually go in and you delete it. So basically, now we're able to use this HPC approaches to enable us to do basically really precise genome surgery and start to think about going in and refolding the genome and to kind of correct disorders. So Power8 has been helping us kind of accelerate workflows from the very beginning. So this process of reading 3D genomes and figuring out what the different structural principles are and figuring out where all the loops are. And then going through and actually writing 3D genomes. So by using GPU-accelerated simulations, we can model the process of loop formation and go in and actually change the structure of the genome. So I'd like to thank my advisor, Erez Lieberman-Aden. He's the director of the Center for Genome Architecture, and also my co-first authors, Miriam Huntley and Adrian Sanborn. And I should mention that so far we've been sharing time on the Rice Power8 cluster, and we've been so happy with it that we're planning on, or we're thinking about getting one of our own as well. So